Barbara McClintock, A Genius Unbound. Part 1. The Quiet Beginnings. A Different Child. Barbara McClintock was born on a warm June day in 1902 in Hartford, Connecticut. The world she entered was one governed by tradition, where women were expected to follow domestic paths. But from the moment Barbara began to walk and talk, she defied expectations. She was quiet, curious, and happiest when alone with her thoughts. Her mother, Sarah Handy McClintock, often remarked to friends, "Barbara doesn't chatter like the other children, she listens to the wind." Her father, Thomas Henry McClintock, a homeopathic physician, recognized early on that his youngest daughter was destined for something extraordinary. In school, Barbara's teachers found her difficult to place. She didn't laugh at childish jokes or crave attention. Instead, she devoured books, especially on science. By age 10, she could name bones, muscles, and had a deep interest in how life worked. When she was 11, her family moved to Brooklyn, New York. It was there, in the bustling energy of city life, that Barbara began to isolate herself, not out of fear, but from purpose. She spent hours at the Brooklyn Botanical Garden, watching insects pollinate, sketching plant life, observing soil, and imagining invisible patterns connecting everything. Barbara did not seek praise. She sought answers. A woman at Cornell. By 1919, Barbara entered Cornell University's College of Agriculture. While women were allowed in the program, they were barred from pursuing majors like genetics at the graduate level. Barbara didn't care. She enrolled in botany, but quickly found her way into the maize, corn, cytogenetics laboratory. Maize was a perfect subject. Its chromosomes were large and easy to view under a microscope, and its colorful kernels reflected genetic traits with surprising clarity. Barbara soon began helping graduate students and eventually leading her own experiments. She worked relentlessly. Others partied on weekends. She stayed in the lab, sketching chromosome patterns, mapping crossbreeding traits, and challenging professors twice her age. One early advisor, Dr. Rollins Emerson, was astonished by her precision. "She sees what others miss," he told a colleague, "and she asks questions that unsettle our assumptions." Despite institutional barriers, Barbara pushed through. In 1927, she received her Ph.D. in botany from Cornell, but by then she was already more accomplished than many faculty members. The language of chromosomes. Barbara began to see chromosomes not as static structures, but as dynamic entities with agency. She believed they held a kind of logical language. In maize, she noticed that chromosomes didn't always behave predictably. Sometimes, traits would suddenly appear or disappear across generations, as if genetic rules had been rewritten. This led her to suspect that genes weren't fixed on the chromosome like beads on a string. Instead, they might move. Her colleagues dismissed the idea. "Ludicrous," said one, "genes can't jump." But Barbara had proof. She found patterns in chromosome breakage, especially in regions she called knobs and centromeres. She developed staining techniques to track these shifts in real time and showed visually how genetic elements could indeed transpose. She wasn't just discovering gene movement. She was decoding one of life's most profound secrets, the woman who would not compromise. Throughout the 1930s and 1940s, Barbara McClintock became known for her brilliance, but also her aloofness. She refused to publish prematurely. She scoffed at fame. She worked independently, often declining co-authorship, and she loathed the scientific culture that demanded performance over integrity. At Cold Spring Harbor Laboratory, she finally found a home. The lab gave her the freedom to explore without interruption. There, surrounded by pine trees and coastal fog, she disappeared into maize, gene maps, and the sacred language of DNA long before the double helix was even understood. She often spoke of her corn plants as if they were friends. "They talk to me," she once saw. By 1950, she published her groundbreaking paper describing controlling elements now known as transposons or jumping genes. No one listened. Reviewers called it too speculative. Colleagues dismissed it as curious but irrelevant. Journals refused to publish follow-ups. Barbara didn't protest. She simply stopped publishing. For over 2 decades, she worked in silence, perfecting, observing, recording, but rarely sharing. Rediscovered and revered. In the 1970s, something unexpected happened. With the advent of molecular biology and the discovery of transposable elements in bacteria and viruses, the scientific community suddenly realized Barbara had been right all along. Her work, once scorned, was now revered. In 1983, she received the Nobel Prize in Physiology or Medicine, becoming the first woman to receive the award Solonat shared with men. During her acceptance speech in Stockholm, she said, "I was astonished that anyone was interested. All I ever did was listen to what the maze was telling me." The world had finally caught up with Barbara McClintock. Part 2. Maze as a Messenger. A world in a kernel. To most scientists, corn was just another plant. To Barbara McClintock, it was a universe. Each kernel yellow, purple, speckled, or striped was a message, a living code revealing the orchestration of genes. Corn, or Zea mays, had large chromosomes that could be viewed under a microscope without advanced technology, making it the perfect model for cytogenetic research. Barbara loved maize, not only for its practicality, but also for its elegance. She marveled at its ability to display genetic variability in ways other plants could not. In maize, a single cross-pollination could yield ears with hundreds of kernels, each one a genetic experiment. "You can see the genetics at work," she would say. "It's like watching evolution in real time." While others pursued fame in publishing or prestigious lectures, Barbara spent hours alone in cornfields, documenting crossbreeds, mapping traits, and quietly challenging the foundations of classical genetics. The art of observation. Barbara's methodology was meticulous. She didn't rely solely on theory, she relied on what she observed. She would stain chromosomes with dyes, placing thin slices of maize root under microscopes, tracking every movement and anomaly. It was this devotion to direct evidence that led her to several breakthroughs. In the early 1930s, she discovered crossing over in chromosomesa phenomenon, where segments of chromosomes exchanged places during reproduction. Though others had theorized it, Barbara was the first to visualize it, proving that recombination occurred at the cellular level. Later, she tracked the ring chromosomes, noting that sometimes chromosomes would form a complete circling anomaly that, again, others failed to believe. But Barbara had drawings, slides, and data. Her evidence was unimpeachable. "The chromosomes were my companions," she wrote. "They gave up their secrets slowly but faithfully." Naming the unseen. Barbara's keen eye for chromosomal behavior led her to detect something even more astounding, the presence of what she would later call controlling elements. These were genetic sequences that could influence the activity of nearby gen-essentially DNA that turned other DNA on or off. At the time, geneticists operated under the belief that genes were static unchanging, but Barbara noticed patterns that didn't make sense under this assumption. Some maize kernels developed odd patterns, streaks, patches, or colors where none should be. These anomalies weren't errors, they were messages. Messages that something within the genome was moving. By identifying these controlling elements, Barbara had made the first known discovery of transposable elements, a concept so ahead of its time that it would take more than 20 years for the rest of the scientific community to understand. "The genome is not static," she insisted. "It's a dynamic system responding to its environment, a community of one." While Barbara was pushing boundaries in maize genetics, the world around her remained skeptical. Her colleagues were largely men, many of whom dismissed her ideas as eccentric or even impossible. She wasn't invited to many conferences. Her research proposals were often returned with vague rejections. She didn't complain. She withdrew further into her work, relying only on herself. She kept a small notebook filled with thoughts, doodles, and genetic observations. She recorded every experiment in meticulous detail. She didn't need affirmation. What she sought was truth. At Cold Spring Harbor, she lived in a modest cottage by the sea, rising early each morning to walk among the fields, checking her maze, whispering questions, listening for answers. They thought I was odd, she later joked. But I wasn't odd, I was just focused. Seeds of controversy. In 1950, she published a revolutionary paper on gene regulation in the journal Proceedings of the National Academy of Sciences. The paper described the behavior of AC, activator, and Ds, dissociator, elements to specific transposable sequences in maize. Barbara demonstrated that the presence or absence of these elements could change gene expression. For example, if Ds inserted itself near a gene responsible for kernel pigmentation, that gene could be silenced or altered, producing streaked or colorless kernels. Her findings were not hypothetical, they were demonstrable, documented, repeatable. And yet, few scientists believed her. Some said it was too complex. Others dismissed it as inconsequential. Many simply didn't understand it. I never thought of quitting, she wrote, but I thought of disappearing, a mind unbroken. Rather than argue, Barbara retreated from the spotlight. She ceased publishing widely, choosing instead to work quietly, logging her discoveries, and perfecting her understanding of genetic mobility. This period became known among her colleagues as the silent years. But silence didn't mean inactivity. She expanded her research into plant stress responses, showing how environmental pressures could trigger the movement of transposons. She proposed that genomes had the capacity to restructure themselves to adapt to new conditions, an idea that challenged both Darwinian and Mendelian orthodoxy. To some, it bordered on heresy. To Barbara, it was clarity. The genome is a responsive, interactive system, she said. We are only just beginning to understand how alive it really is. Corn as a mirror. By the late 1960s, as molecular biology took center stage and Watson and Crick's double helix revolutionized the understanding of DNA, Barbara's work began to draw cautious interest. Younger scientists revisited her data, re-examined her slides, and began to see the brilliance buried within her maize kernels. Barbara, now in her 60s, remained unbothered by the belated recognition. She still walked the cornfields every morning, still peered through microscopes, still took her meals in solitude. She had never sought applause. Her joy came from the work. In an interview, she said, "I've never thought of science as a career. I thought of it as something I loved." And love it she did with unwavering passion, solitude, and devotion. Cytogenetics and chromosome logic. The birth of cytogenetics. In the early 20th century, genetics was still in its infancy. Mendel's rediscovered laws of inheritance had just begun to shape biological thought, but the physical basis of gene show and where they were located remained a mystery. It was in this intellectual landscape that Barbara McClintock rose like a solitary flame. Her curiosity about how traits passed from one generation to the next led her to a revolutionary concept that chromosomes were not only carriers of heredity, but active participants in the story of life. Working in the Mays laboratory at Cornell, Barbara began using advanced microscopy techniques and innovative staining procedures to observe the behavior of chromosomes in real time. She didn't just look, she listened. She watched chromosomes during meiosis and mitosis, drawing careful diagrams and annotating observations with obsessive precision. "You must have the patience to sit with them," she once told a student. "They tell you everything if you know how to read." Her drawings were works of art technically perfect, intuitively brilliant. They became some of the first visual maps of chromosome behavior in plants, crossing over and chromosome mapping. One of Barbara's earliest triumphs in cytogenetics came in the form of concrete evidence for crossing over, a term used to describe the exchange of genetic material between paired chromosomes during meiosis. Though crossing over had been hypothesized, Barbara became one of the first scientists to observe it physically under the microscope in maize cells. She matched specific genetic traits with visible structural changes in chromosomes, creating cytological maps, a stunning fusion of physical biology and genetics. She identified the relationship between gene locations and chromosomal structures with such clarity that other scientists were awestruck. She was not merely observing biology, said one colleague. She was decoding its architecture. Her ability to connect the invisible genes to the visible chromosome structures laid the groundwork for the field of cytogenetics, a fusion of cytology, cell biology, and genetics, ring chromosomes, and breakage fusion bridge cycles. While most researchers studied what genes did, Barbara studied what they were. In 1938, she discovered an entirely new phenomenon, ring chromosome-escircular formations that resulted from the fusion of chromosomal ends after breakage. But the discovery didn't stop there. She noticed that once a chromosome broke, it wasn't merely damaged, yet it became unstable, prone to further breakages and fusions in subsequent generations. This observation led to her most complex model yet, the breakage, fusion, bridge, BFB cycle. Here's how it worked. A chromosome breaks. The broken ends fuse, forming a ring. During cell division, the ring chromosome is torn apart unevenly. This leads to further breakage and fusion. Over time, this generates genetic instability and variability. This model provided the first mechanistic insight into how genetic instability could be propagated across cell generations, a concept foundational to modern understanding of cancer genetics. The genome is not immutable, she wrote. It changes, reorganizes, and evolves. Today, BFB cycles are recognized as key contributors to tumor evolution and chromosomal disorders. The power of detail. Barbara's discoveries weren't accidents. They were the results of relentless discipline and detail. She didn't rely on computer models or mass data; those didn't exist yet. She relied on her eye, her microscope, and her journal. Every slide she prepared was a story. Every kernel was a clue. Every anomaly was an opportunity to ask why. She was known to work 16-hour days in near silence, focused not on producing papers, but on understanding the truth at the chromosomal level. Some of her contemporaries joked that she had a sixth sense for chromosome behavior. But it wasn't magic. It was immersion, total, absolute immersion. You don't force the chromosomes, she once told a researcher. You let them reveal themselves. This philosophical approach of letting nature teach would become her hallmark and a defining principle in experimental genetics. Mentorship and isolation. While Barbara didn't supervise many students, those she did mentor were profoundly influenced. She taught them not through lectures, but by example. They learned to observe before theorizing, question every assumption, never rush an experiment, respect the organism, not just the outcome. Yet, as her ideas grew more complex and abstract, Barbara increasingly found herself alone. Many male scientists, particularly those in molecular biology, dismissed her work as outdated. They preferred chemical explanations and gene sequencing, ignoring the architectural and structural elements she had uncovered. She became a ghost in her own field, referenced, but rarely cited. She didn't play the academic game, said one contemporary. She played the long game, and she won. The genome as a responsive system, Barbara's theories began to converge around a radical idea, that the genome was not a passive repository of information, but an active, self-regulating system. This went against the dominant thinking of the time, which treated genes as fixed blueprints. Barbara saw the genome as a dynamic entity, one that responded to stress, environmental stimuli, and internal instability. Through her work with maize, she showed how certain elements could jump or transpose in response to damage. Genes could be turned on or off by mobile elements. Chromosomes could restructure themselves to survive. In essence, she proposed a genetic feedback loop system in which the genome could sense, respond, and adapt. "There's a wisdom in the genome," she wrote. "It knows how to protect itself." This perspective foreshadowed epigenetics, gene silencing, RNA interference, and genomic plasticidal concepts that would emerge decades later. The resistance of the scientific establishment. Despite her profound discoveries, the scientific community was slow to accept McClintock's insights. Her work didn't fit the reductionist mindset that dominated post-war biology. Her languageric with metaphors and philosophical observations was often misunderstood as mystical or imprecise. Reviewers rejected her papers. Conference organizers overlooked her. Younger scientists, indoctrinated by central dogmas, ignored her, and yet she never changed. "I do not need to be believed," she once told a colleague. "I need only to understand." In the silence, she flourished. Untethered by the need for approval, Barbara went deeper, connecting chromosomal instability to plant development, to mutation, to adaptation, and even evolution. Her notebooks from this period are now viewed as masterpieces of biological thought decades ahead of their time. The foundation of modern genetics. Today, the impact of Barbara McClintock's work is nearly immeasurable. Her discoveries have influenced cancer biology, BFB cycles, and genome instability are central to understanding tumor progression. Gene therapy. Transposons are now used as vectors to insert genetic material into human cells. Agricultural genetics. Her work with maize laid the foundation for crop improvement, genetic mapping, and hybridization. Epigenetics. Her insights into gene regulation anticipated whole fields of study on gene silencing and activation. She didn't just study chromosomes, she redefined them. She didn't just discover jumping gene, she jump-started an entirely new era of biology. Part 4. Discovery of Transposition. A puzzle inside a kernel. By the early 1940s, Barbara McClintock had already established herself as a leading expert in maize cytogenetics. But her most groundbreaking discovery, the one that would redefine the very nature of genetics, was still ahead. In her observations of maize kernels, Barbara noticed something puzzling. Some ears of corn produce kernels with irregular color patterns, streaked, spotted, or completely devoid of pigment, where color should have been consistent. These changes weren't random. They followed subtle but persistent patterns that pointed to something deeper than simple Mendelian inheritance. To most, they were anomalies. To Barbara, they were signals. "Every kernel was a message," she once said. "All you had to do was learn the code." This code, she believed, pointed toward the presence of genetic regulatory elements within the genome that could influence, suppress, or activate genes in a way that had never been imagined. The AC and Ds elements. In a series of meticulous experiments, Barbara identified two crucial elements in the maize genome. Ds, dissociator, AC, activator. Ds was a chromosomal segment that caused the pigment gene to break, resulting in colorless or spotted kernels. But Ds did nothing on its own. It required the presence of AC to activate the breakage. Without AC, Ds remained silent. This interaction was key. Barbara had stumbled upon the first documented example of genetic elements that move. Not just static segments of DNA, but mobile, responsive sequences that could jump from one chromosome location to another. She called them controlling elements. Later, the scientific world would know them as transposons, or jumping genes. They are dynamic, Barbara explained. They move, interrupt, silence, or initiate the activity of neighboring genes. These findings weren't just intriguing, they shattered the dominant belief that the genome was a fixed, stable structure. For the first time, a mechanism had been observed that could rearrange the genome from within, demonstrating the movement. To prove transposition, Barbara undertook some of the most detailed genetic mapping ever conducted. She traced every movement of AC and Ds across generations, linking changes in phenotype, like kernel color, to chromosomal changes. She developed a system of cytological genetic correlation, where visual chromosome changes matched genetic expression shifts. She could predict with eerie accuracy where AC or Ds had moved and what effect they would have. Her work revealed that AC could increase in copy number, multiplying its influence. Ds could insert itself into coding genes, silencing them until AC activated it. The timing of AC activity could lead to mosaic patterns in kernels, some parts pigmented, others colorless. These patterns were not noise. They were biological poetry, precise, expressive, and evidence of an orchestrated genomic dance. Transposition under stress. One of Barbara's most profound insights was the relationship between environmental stress and transposable activity. She hypothesized that under conditions of stress such as radiation, temperature shifts, or disease-transposable elements were more likely to activate, reorganizing the genome as a survival response. This was nothing short of revolutionary. It implied that the genome was not only mobile, but intelligent, capable of responding to threats by activating internal mechanisms of adaptation. A genome can reorganize itself, Barbara wrote, not as a passive target, but as an active agent. This theory challenged evolutionary biology, which traditionally relied on random mutation and natural selection. Barbara was proposing an inbuilt genomic flexibility and idea decades ahead of its time. Today, this concept is foundational to the field of epigenetics and genomic plasticity, the 1,950 paper and the silence that followed. In 1950, Barbara McClintock published her seminal paper, The Origin and Behavior of Mutable Loci in Maize, in Proceedings of the National Academy of Sciences. The paper described, in astonishing detail, the discovery of AC and Ds, their transposition behavior, and the larger implications for genetic regulation. The response? Silence. Many of her peers found the work too speculative or too difficult to follow. Some dismissed her terminology. Others could not fathom the idea of genes that moved. At the time, the concept of regulatory DNA was not yet mainstream. DNA itself had not been structurally characterized. It would be another 3 years before Watson and Crick proposed the double helix. Barbara had leaped a generation ahead. No one was ready, she later recalled. I think they were frightened by the implications. Disheartened by the lack of engagement, Barbara made a bold decision. She stopped publishing on the topic. Instead, she turned inward, continuing her work in silence, refining her observations, and trusting that the maze would reveal more in time. Personal reflection and inner conviction. The failure of the scientific community to grasp her discoveries didn't break Barbara's spirit, it deepened her resolve. She spent the 1950s and 1960s in quiet reflection at Cold Spring Harbor. She worked alone, kept extensive notebooks, and rarely attended conferences. Many of her colleagues believed she had abandoned science. In truth, she was letting science unfold at its own pace. She also began to write more philosophically, exploring the relationship between the observer and the organism. She rejected the idea that science was a conquest of nature. To her, it was a dialogue. You don't impose your will on a living system, she said. You listen. This attitude set her apart in an era of growing technological arrogance. As scientists rushed to manipulate genes, Barbara remained a steward seeking to understand, not to control. Redemption in the molecular age. As molecular biology matured in the 1970s, scientists began discovering transposable elements in other organisms, bacteria, fruit flies, even humans. Suddenly, Barbara's work seemed not just relevant but prophetic. Geneticists found that transposons influenced antibiotic resistance in bacteria. Mobile elements regulated development in Drosophila, fruit flies. Genomic rearrangements could lead to cancer or autoimmune diseases in humans. The field had caught up to Barbara McClintock. By the early 1980s, researchers around the world were citing her 1,950 paper with reverence. She was invited to give lectures, honored by universities, and nominated for the highest scientific awards. And then, in 1983, came the ultimate recognition, the Nobel Prize. At the age of 81, Barbara McClintock was awarded the Nobel Prize in Physiology, or Medicine, becoming the first woman to win the prize unshared. The citation read, "For her discovery of mobile genetic elements." In Stockholm, she delivered an understated but moving speech. She spoke not of glory, but of curiosity, not of triumph, but of gratitude. She thanked the corn plants. She thanked the genome. "I was never alone," she said. "The maze was with me." The room erupted in applause. For decades, the world had ignored her. Now it bowed in admiration. A legacy written in code. Barbara's discovery of transposable elements redefined genetics, launching new fields of inquiry: functional genomics, gene regulation, precision medicine, evolutionary adaptability. Today, transposons are recognized as occupying nearly half of the human genome. They play roles in gene expression, immune response, neural development, disease progression. Barbara didn't just discover something new. She revealed that the genome was alive, dynamic, adaptive. In doing so, she redefined what it means to understand life. Part 5. The Silent Years. The Decision to Vanish. After her groundbreaking 1,950 paper on transposable elements, Barbara McClintock found herself at an unusual crossroads. She had delivered one of the most paradigm-shifting scientific revelations of the century and was met with silence, or worse, dismissal. While many scientists would have responded with defiance, publication campaigns, or institutional lobbying, Barbara chose the opposite path. She retreated. In the eyes of her peers, she had disappeared. In truth, she had turned inward, where the real work was waiting. She stopped publishing. She declined conference invitations. She stopped seeking grants. Instead, she spent her days walking the fields at Cold Spring Harbor, observing her maze, making notes in thick brown journals, and sketching chromosomes with pencil and ink. "I didn't leave science," she later said. "I left the noise." Cold Spring Harbor, a monastery of thought. Barbara's refuge during these decades was the Carnegie Institution's Department of Genetics at Cold Spring Harbor, nestled along the north shore of Long Island, New York. The campus wind-swept, tree-lined, and far from urban distractions became her sanctuary. While others chased grants and tenure, Barbara practiced science like a meditative ritual. Each morning she awoke early, visited the greenhouses, checked on her maze, and returned to her modest cottage to write, read, and think. She lived, simply never married, never pursued material luxury. Her home was filled with books, field samples, and microscope slides. She rarely socialized, but welcomed anyone willing to talk earnestly about science, nature, or philosophy. "I have no time for pretense," she once told a young biologist. "If you want to learn, come and observe. Listen with your whole mind." Conversations with Mays. In solitude, Barbara refined what she called her dialogue with the genome. She didn't view plants as objects of manipulation, but as collaborators in knowledge. The maze would speak, and she would listen. Its patterns, mutations, irregularities all told a story. She believed that to truly understand a living system, A scientist must become immersed in it not from above, but alongside. This was not mysticism, it was empathy as method. You must have a feeling for the organism, she explained in a rare interview. You must understand its nature, its behavior, its purpose. Only then will it reveal its secrets. She began documenting not just scientific outcomes, but philosophical insights. Her journals from the 1950s, 1970s read like a fusion of biology, poetry, and epistemology. A watcher of change. During the silent years, Barbara continued experimenting, though she rarely shared her results. She observed how transposable elements responded to stress, drought, heat, viral infection. She recorded how the genome adapted, repaired, and protected itself. She suspected that the genome was capable of structural self-awareness. It could reprogram itself in response to threats. It had embedded intelligence in the conscious sense, but in adaptive logic. These were staggering ideas for her time. They bordered on heresy in a scientific world built on predictability and linear causality. She didn't care. "I know what I see," she wrote. "If no one else sees it yet, they will eventually." The visitor from Yale. In 1978, a graduate student from Yale named Evelyn Fox Keller visited Barbara to interview her for a biography. She expected to meet a bitter recluse. What she found instead was a serene master, wholly at peace, intellectually luminous, and deeply reflective. Their conversations revealed a philosophy of science unlike anything Keller had encountered. That scientific discovery was not about conquering nature, but aligning with it. That true understanding required humility. That the ego was the enemy of insight, Keller was struck by Barbara's refusal to see her life's work as vindication or tragedy. To Barbara, truth was never measured by fame, but by integrity of observation. The eventual book, A Feeling for the Organism, brought Barbara back into public view. But more importantly, it articulated a new model for scientific thinking, one driven by empathy, intuition, and patience. Recognition begins to bloom. By the late 1970s, molecular biologists began to discover transposons in bacteria, viruses, and fruit flies. Suddenly, Barbara's controlling elements weren't bizarre outliers, they were universal. Young researchers pored over her 1,950 paper with astonishment. It had all been the mechanics, the predictions, the insights waiting for the tools and language to catch up. Conferences began inviting her again. Universities began awarding her honorary degrees. The scientific community had turned, humbled, to its silent pioneer. "She didn't change," said one researcher. "We did." A legacy reawakened. Cold Spring Harbor began archiving Barbara's papers. Students arrived in busloads to hear her speak. Her notebooks, once private sanctuaries, were now treasured artifacts of scientific history. Even those outside genetics philosophers, ecologists, feminist scholars, began studying her as a model of intellectual independence and scientific integrity. Barbara welcomed the attention but remained unmoved by it. She often quoted one of her favorite lines, "You must never stop working, even if no one listens. Nature is always listening." The global genome. In the years before her Nobel Prize, Barbara's influence quietly expanded across disciplines. Agriculture. Her maize work laid the groundwork for crop mapping and hybridization techniques. Medicine. Her theories of genome instability informed cancer biology. Biotechnology. Transposons became tools for gene insertion and therapy. Evolutionary theory. Her work challenged the slow, random mutation model of evolution, and perhaps most poetically, her insights about genomes responding to stress anticipated the field of epigenetics, where gene expression is altered not by mutation, but by environmental feedback. Barbara had seen this decades earlier. She simply didn't have the language yet, but the maze had told her, and she had listened. Peace in solitude. By the time she was in her late 70s, Barbara McClintock had become an icon by design but by virtue. She continued to live alone, take her walks, visit the maze, and write in her journals. She never installed a telephone in her cottage, preferring silence to distraction. She taught young scientists that a laboratory was not a factory but a sanctuary, that curiosity was a sacred thing, and that the pursuit of knowledge should never be tainted by ego or reward. The more you know, she told one student, the more you realize how little you control. The key is to remain open. The end of the quiet years. Though she never returned to formal academic roles, Barbara's final years before the Nobel were anything but idle. She corresponded with researchers around the world, quietly guiding new generations of geneticists and biologists. In the spring of 1982, she was invited to a scientific symposium at MIT. The room was packed with hundreds of researchers. She stood before them not as a relic, but as a visionary. And in a soft voice, she shared the same message she had lived by for half a century. Watch closely, ask gently, let the organism tell you the story. Part 6. The world catches up. A world wakes up. In the early 1980s, decades after Barbara McClintock quietly laid the groundwork for understanding the dynamic genome, the biological world began to stir. Young molecular biologists armed with new technologies were making discoveries that mirrored what Barbara had already seen in maze through her microscope and intuition alone. In laboratories across the globe, scientists were sequencing mobile genetic elements in bacteria, observing gene silencing and activation in Drosophila, fruit flies, linking genome instability to human diseases like cancer and immunodeficiencies. The term transposon, once obscure, became a fixture in genetic literature, and slowly papers began citing Barbara's 1,950 publication with reverence. Her name, long spoken only in small circles, now appeared in textbooks and conferences. She wasn't just a pioneer, one scientist noted. She was the first one to step onto a new continent and draw the map. The epigenetics revolution. One of the most dramatic vindications of Barbara's work came from the emerging field of epigenetics, the study of how genes are expressed or silenced without altering the DNA sequence itself. Barbara had long observed that genes could be turned on or off based on their chromosomal environment, especially when influenced by transposable elements. This challenged the prevailing assumption that DNA sequences alone determined biological destiny. As scientists began to uncover histone modifications, DNA methylation, non-coding RNAs, they realized what Barbara had intuited decades earlier. The genome was a fluid, adaptive, and context-sensitive system. In a 1983 symposium on epigenetics, one molecular biologist concluded his lecture with a simple slide. Barbara was right. Biotechnology embraces the jumping gene. Another sign that the world had caught up was how transposons began to serve practical purposes in biotechnology and medicine. Researchers found that transposons could be engineered to deliver genes into living cells, a technique vital to gene therapy. Mobile elements played roles in the adaptive immune system, allowing cells to shuffle gene segments to produce antibody diversity. Genome editing tools like CRISPR benefited from principles first revealed by studying transposons. From agriculture to pharmaceuticals, Barbara's discoveries became instruments of innovation. Biotech companies began developing transposon-based delivery systems. Plant breeders used her maze maps for hybrid optimization. Medical researchers examined transposon activity in neurodevelopmental disorders. Barbara, once ignored, had become a keystone of modern biology, rejoining the scientific conversation. Though she never actively sought the spotlight, Barbara found herself pulled back into it as the 1980s advanced. Invitations from leading universities poured in. Scientific societies, journals, and government agencies wanted her to speak, consult, or receive awards. She accepted some, but always on her own terms. At one such gathering, a young postdoctoral student nervously approached her. "Professor McClintock, I've read everything you've written. Your work changed my life." Barbara smiled gently and replied, "Then you have Mays to thank." Awards and honors. As the full scale of her contributions became clear, Barbara began receiving the recognition that had long eluded her. Year Award honor, 1,970 National Medal of Science, U.S., 1,981 Albert Lasker Award for Basic Medical Research, 1,982 Wolf Prize in Medicine, 1,983 Nobel Prize in Physiology or Medicine, 1,985 inducted into the National Women's Hall of Fame, 1,986 Genetics Society of America Medal, 1,987 elected foreign member of the Royal Society, UK. The accolades, however, never changed her. When reporters asked her how it felt to finally be recognized, she answered with disarming honesty, "Recognition is nice, but it's never been my goal. I've always just wanted to understand." A feeling for the organism. Perhaps the most significant tribute to Barbara McClintock's legacy came not from a medal, but from a book. In 1983, Evelyn Fox Keller's biography, A Feeling for the Organism, was published. It portrayed Barbara not only as a scientist, but as a philosopher woman whose empathy, intuition, and independence had redefined how science could be done. The book captured Barbara's unique approach, listening to her subject rather than controlling it, respecting complexity over simplification, trusting deep attention over quick results. It inspired not only geneticists, but educators, philosophers, and women in science. In classrooms across the U.S., students read the story of a quiet genius who followed her own rhythm and changed the world. Role model for women in science. Barbara's life also became a rallying point for women in science, a reminder of how brilliance and persistence can endure even in hostile environments. She had never branded herself a feminist, but her life embodied the struggle and triumph of women in a male-dominated field. She faced systemic discrimination, institutional skepticism, cultural isolation, and she prevailed without compromising her methods, her temperament, or her ideals. Young women began writing to her. Science teachers shared her story in lectures. Universities cited her life in promoting gender equity in STEM. Barbara often said little about it publicly, but privately she expressed pride. "If I've helped others stand their ground, then I've done something worthwhile." The genome's guardian. As the 1980s drew to a close, Barbara continued working, albeit slowly. Her eyesight was fading, her steps less steady, but her mind remained razor-sharp. She mentored, corresponded, and reflected. She viewed the genome as a living story, one still unfolding, one that humanity had only just begun to read. She believed that the next generation of scientists would not only decode life, but learn how to respect it. She hoped they would. Observe with care, ask with humility, protect what they came to understand. To the very end, Barbara McClintock was not just a scientist, she was a guardian of the living code, the moment the world knew. At the Nobel Prize ceremony in Stockholm, Barbara stood before the King of Sweden and the scientific elite of Europe. Her speech was short. She didn't thank committees or institutions. She didn't grandstand. Instead, she told a story of Mays, a story of patient listening, a story of how a genome could speak if one knew how to listen. "We have to learn to see with the eye of the genome," she said, "to feel with its processes, to think with its movement." In that moment, the world didn't just honor a scientist. It understood her. Part 7, Nobel and Beyond. Stockholm, 1983. On the 10th of December, 1983, Barbara McClintock stood beneath the grandeur of the Stockholm Concert Hall. Dressed in a simple dark gown with her signature spectacles perched firmly on her nose, she approached the stage to accept the Nobel Prize in Physiology or Medicine, the highest honor a scientist could receive. At 81 years old, she had become the first woman to win the prize unshared, and only the third woman in history to win it at all in this field. The moment was historic, but Barbara treated it as simply another step in a lifelong conversation with nature. "I have not changed," she told a reporter the day before the ceremony. "Only the world has begun to listen." Her speech was brief, humble, and deeply personal. She gave credit not to herself, but to Maser, companion in exploration, and to the genome, which had revealed its secrets not all at once, but through years of patient, devoted observation. The quiet after the applause. The Nobel Prize brought Barbara global fame. Her face appeared on magazine covers. She was interviewed by journalists, featured on television, and honored at banquets across the world. But for Barbara, all of this was noise. She returned to Cold Spring Harbor immediately after the ceremony, where she resumed her routine of long walks through the fields and evenings spent reading and reflecting in solitude. She responded politely to interview requests, but rarely accepted them. She turned down speaking engagements unless they were meaningful. She answered letters from young scientists with warmth, but always with brevity. "She never let the prize define her," said one colleague. "She had nothing to prove before and nothing to defend after." The philosopher of the genome. In the years that followed, Barbara began to write more reflectively about the meaning of her discoveries. Her late essays revealed a mind that was not only scientific, but philosophical. She believed that science must remain open to mystery, that no matter how much data one gathered, true insight required something deeper, the ability to feel the rhythm of life itself. She argued that the genome was not a machine, but a system in motion. Genes didn't simply encode traits, they responded, interacted, and transformed. Scientific inquiry should be guided by intuition, patience, and empathy with the organism. These writings influenced not only biologists, but thinkers in systems theory, ecology, and feminist science studies. Her voice became one of quiet authority, a reminder that discovery is not merely about control, but about communion. Guardian of a legacy. Barbara had no children, no spouse, no heirs in the traditional sense. But she left behind a living legacy, the hundreds of scientists, students, and thinkers inspired by her life and work. At Cold Spring Harbor, her lab was preserved. Her journals, slides, and notes were archived and studied. Her former students became professors, authors, and researchers who passed on her philosophy. Even in institutions that once ignored her, her name now adorned. Lecture halls, research fellowships, awards in genetics and cytogenetics. She became a patron saint of curiosity, a woman whose independence became a model for anyone daring to walk alone in pursuit of truth. "She taught me not how to study genetics," one mentee said, "but how to be a scientist." Final years of reflection. In her final years, Barbara slowed her pace. She spent more time at home, surrounded by books and wildflowers. She still took occasional walks to the maize plots, though she no longer performed experiments. She wrote a final letter to a close friend saying, "The genome has more to teach us yet. Perhaps one day we will understand it not just as code, but as consciousness." She remained healthy, sharp, and full of wonder until the very end. On the 2nd of September, 1992, at the age of 90, Barbara McClintock passed away quietly in Huntington, New York. There were no headlines that day, no breaking news, but in labs and classrooms around the world, a quiet moment of reverence passed. After the end. Today, Barbara's legacy endures in every gene therapy, every crop map, every DNA regulation study. Her work laid the groundwork for, CRISPR technologies, genome sequencing, epigenetic therapeutics, adaptive systems theory. She is recognized not just as a discoverer of transposons, but as a founder of modern genetics. Her portrait hangs in museums. Her name is etched in marble. But more importantly, her approach, careful, intuitive, respectful, has found its way into the very culture of scientific inquiry. "I was never alone," she once said. "I was always in conversation with maize, with chromosomes, with life itself." Part 8. Personal Life and Philosophy. A life chosen, not inherited. Barbara McClintock's personal life was, in many ways, as unconventional as her scientific journey. She never married, never had children, and never lived with a partner. To some observers, her life may have seemed solitary, almost monastic. But to Barbara, it was intentional, a choice born from conviction, not deprivation. "I couldn't have done this work with distractions," she once said. "Science demands your whole being." While many of her female contemporaries were expected to balance marriage and motherhood, with careers often sacrificing one for the other, Barbara bypassed the struggle entirely. She belonged wholly to her work. She made no apologies for her independence, nor did she seek sympathy for the solitude. Her relationships with plants, notebooks, and the natural world were rich and fulfilling in their own right. The power of silence. Barbara valued silence, not just the absence of sound, but the psychological space in which thought could grow. At Cold Spring Harbor, her cottage was filled with peace. No television, no telephone, no constant visitors. She once remarked that too much noise diluted intuition. Her silence was not withdrawal, it was attunement. To see what others missed, she needed the quiet to hear what the maze was saying. "The answers are always there," she wrote, "but only when you learn to ask the right questions in the right way." Feminism without labels. Though Barbara McClintock is now revered as a feminist icon, she never labeled herself as such during her lifetime. She avoided political debates and steered clear of identity-driven narratives. Her focus was purely on the work, and yet her very existence as a fiercely independent, brilliant woman in a male-dominated field became a statement. Her refusal to compromise, to shrink, to conform was activism in action. She carved a path for women, not by campaigning, but by demonstrating what was possible when brilliance was left unshackled. "I never thought of myself as a woman scientist," she once said, "just as a scientist." To generations of female researchers who followed, Barbara became a beacon not because she fought for space, but because she occupied it unapologetically. A spiritual scientist, while Barbara was not religious, she often spoke about nature in reverent terms. Her relationship with the living world had a spiritual texture. She believed in wholeness, that organisms must be understood as complete systems, not broken into parts. Emergence, that life's properties could not always be predicted from its component. Respect, that the scientist's role was not to dominate nature, but to learn from it. Her idea of a feeling for the organism wasn't sentimentality, it was method. An epistemology rooted in presence, care, and humility. She once said, "There is something deeply wise in the genome. It is not just codate, it is history, memory, response, and possibility." Her view foreshadowed ideas in systems biology, complexity theory, and ecoethics decades before those fields existed. Letters from the young. In her later years, Barbara began receiving hundreds of letters from students, especially young women. Some were scientists, some were aspiring, some simply admired her courage. They asked, how did she persevere in isolation? How did she know her work had value when no one else did? How did she stay true to herself? Barbara answered many of them, often by hand. Her responses were typically short, but always thoughtful. She encouraged them to stay curious, be patient, trust observation, avoid arrogance, and most of a listen, what she believed. Barbara McClintock's philosophy wasn't written as a manifesto. It was lived in her choices, her experiments, her rhythm. But if one had to distill it, her beliefs might include that nature is more subtle than we realize. That scientific insight requires both rigor and intuition. That isolation can be fruitful if one listens deeply. That truth doesn't demand belief, it just is. That science, done properly, is a form of love. She never sought followers, but in living authentically, she inspired thousands. The ritual of work. For Barbara, scientific work was not a task, it was a ritual, a sacred rhythm. Her daily life was simple: wake at sunrise, walk to the maize plots, observe the leaves, the soil, the wind, take notes, sketch chromosomes, reflect, repeat. She did this not for reward, but because it was who she was. This ritual kept her grounded. It connected her to something timeless, something beyond fame, beyond failure, beyond human noise. I never felt alone, she said. In the field, I was with the maize. In the lab, with the chromosomes. In the silence, with the genome, the language of living things. Barbara's deepest insight may not have been scientific at all, but epistemological. The way she believed we come to know things. She saw the genome not as a static book, but as a living textstone that changed, adapted, and sometimes rewrote itself in response to the world. To read it properly, one needed more than technique. One needed respect. Her work, then, was not about conquering knowledge, but receiving it. With open hands, with quiet eyes, with humility. In this way, Barbara McClintock became not just a discoverer of facts, but a teacher of ways of knowing. Part 9. Legacy in Science and Feminism. The seed that changed biology. Barbara McClintock may have worked with maize, but her legacy would grow far beyond the cornfields of Cold Spring Harbor. Her discovery of transposable elements did more than alter the course of genetic research. It redefined the nature of the genome itself. From her quiet cottage and microscope came a revolution that touched every corner of life science, from agriculture to neuroscience, from virology to biotechnology. Her vision of the genome as responsive, fluid, and self-regulating became the foundation of epigenetics, functional genomics, Generegulation-Studies, evolutionary developmental biology, precision medicine. She showed us that the genome is not a frozen blueprint, one geneticist later said, but a living manuscript that rewrites itself. Geneticist of the future. By the early 2000s, the Human Genome Project was nearing completion. Researchers had mapped the sequence of over 3 billion base pairs in the human genome. But what astonished them was this. A large portion of the genome consisted of transposable elements, precisely the type of DNA Barbara McClintock had identified over half a century earlier. Far from being junk DNA as once presumed, these elements were found to influence gene expression, regulate developmental timing, play roles in neurological and autoimmune diseases, contribute to immune system adaptability. Barbara hadn't merely discovered an oddity. She had found the architectural grammar of genomes. Her work continues to inform gene editing technologies like CRISPR-Cas 9, synthetic biology, and artificial genomes, cancer genome instability research, stem cell and regenerative medicine. In essence, she was a scientist of the 21st century living in the 20th, the woman who waited. Barbara McClintock's story has also become a powerful case study in scientific perseverance. She waited over 30 years for her work to be understood. She did not waver. She did not rush. She did not dilute her findings for easier consumption. In doing so, she redefined what it meant to succeed in science and know through popularity or publications, but through clarity and conviction. Her story is now taught in science courses as a lesson in resilience, vision, the cost of being ahead of one's time. She didn't bend to the world, one scholar wrote. She waited for the world to bend toward the truth. Scientific integrity in practice. In an age where metrics, funding, and fast publication often dominate academic life, Barbara's approach remains a model of scientific integrity. She refused to publish prematurely, withdrew when she felt her work was misunderstood, shared ideas only when fully tested, avoided self-promotion. This has inspired a new generation of scientists who advocate for slow science, deep observation, ethics in research, reproducibility, and transparency. Her method was not just about what she studied, but how she studied. A feminist icon without saying so. Though she never embraced the label, Barbara McClintock's life resonates deeply with feminist scholars and women in STEM. Why? Because she refused to conform to male expectations, created her own path in a system not built for her, held her ground with brilliance instead of bravado, proved that success didn't require compromise. She was a woman who defined herself not in opposition, but on her own terms. Today, her story is used in women's studies curricula, STEM inclusion programs, leadership development seminars for women in academia. Her legacy reminds aspiring female scientists that there is power in quietness, strength in patience, and brilliance in authenticity. Barbara McClintock didn't fight for a seat at the table, wrote one author. She built her own lab, her own chair, her own language, remembered by her peers. Following her death in 1992, tributes poured in from across the globe. James Watson, co-discoverer of DNA's double helix, called her one of the most original and brilliant minds in genetics ever. Françoise Jacob, Nobel laureate, said, "McClintock changed how we understand evolution." Leading journals ran obituaries that read like scripture. The New York Times wrote, "She listened when no one else did, and in listening, she changed the way we read the book of life." Monuments, museums, and memory. Over time, Barbara McClintock's image has adorned U.S. postage stamps, university murals, public sculptures, and science exhibits. Institutions have named awards after her. The McClintock Prize for Plant Genetics, the Cold Spring Harbor McClintock Fellowship, Barbara McClintock Building at the University of Missouri. She has become not only a scientific figure, but a cultural icon symbolizing independence, excellence, and foresight. A lesson for all thinkers. Barbara's influence extends beyond biology. Her life offers lessons for writers in the discipline of quiet observation, philosophers in embracing uncertainty, educators in nurturing patient curiosity, artists in honoring unseen rhythms. She taught that the deepest insights often come from listening instead of speaking, watching instead of rushing, trusting instead of proving. Her model is timeless not only for scientists, but for anyone who seeks truth in a noisy world. What she leaves behind? Barbara McClintock left no heirs in the traditional sense, but she seeded generations of scientists and thinkers. Her impact lives on in the genome editing tools used in hospitals, the cornfields of agricultural researchers, the textbooks of biology classrooms, the dreams of every girl who wonders if science is for her. She is not simply a historic figure. She is a presence quiet, steady, unwavering, reminding us that genius doesn't need permission to bloom. Part 10. Final Years and Eternal Echoes. A Life in Full Bloom. By the 1980s and early 1990s, Barbara McClintock had become a living legend, something she never sought but humbly accepted. From her modest home in Cold Spring Harbor, she watched as generations of scientists finally embraced the truths she had uncovered decades earlier. Despite failing eyesight and aging joints, she never stopped working. She continued writing, mentoring, and walking through the maize fields as long as her body allowed. "I want to be with my corn until the end," she often said with a smile. Her life remained unchanged. She wore the same practical shoes, ate simple meals, and lived surrounded by notebooks, botanical drawings, and microscope slides testaments to a life immersed in discovery. The last conversations. In her final years, visitors came from all over the world: students, professors, journalists, even philosophers. They didn't come for formal interviews, they came for wisdom. Barbara received them in her small living room, always offering tea, always listening more than speaking. She talked about the poetry of the genome, the ethics of genetic manipulation, the danger of ambition without understanding, the importance of feeling for the organism. She reminded each visitor that truth takes time and that the best science, like the best lives, unfolds slowly. Letters to the future. Before her death, Barbara wrote dozens of private letters to young scientists, colleagues, and institutions she believed would shape the future of genetics. Some of them were kept private, others were later published in books and journals. In one, she wrote, "Do not hurry. Let nature show you her rhythm. The truth is not afraid of time." In another, "If you must choose between being understood and being true, choose truth. Understanding will come." These letters became a kind of informal will not of possessions, but of principles. The 2nd of September, 1992, on the morning of the 2nd of September, 1992, Barbara McClintock passed away peacefully in Huntington, New York. She was 90 years old. There were no grand headlines, no national days of mourning. But in labs, libraries, and universities across the world, the news rippled like a wave of reverence. Scientific journals published full-page tributes. Conferences held moments of silence. Former students wept over microscopes. "A great light has gone out," said one Nobel laureate, "but she leaves a path that glows for generations to follow." The genome remembers. Barbara once described the genome as a memory machina system that carried not only genetic instructions, but the history of how life adapted, responded, and survived. Now, it was her own memory that lived unetched into the science she had shaped. Her influence could be found in every medical breakthrough based on gene regulation, every agricultural innovation derived from maize genetics, every child whose disease was treated through gene therapy, in labs using CRISPR, in software mapping genomes, in academic programs, training the next generation of geneticists. Barbara was there. McClintock in the classroom. Today, Barbara McClintock's life is taught not only in biology courses, but also in philosophy of science programs, gender studies classes, history of medicine and ethics seminars. Students read A Feeling for the Organism and watch documentaries about her discoveries. But more importantly, they study how she thought. They learn that silence can be strength. Solitude can be generative. Observation can lead to revolution. Professors across disciplines use Barbara as a case study in intellectual courage, a lesson in trusting one's perception even when the world looks away. Portraits, poems, and plays. Barbara McClintock has inspired not just scientists, but artists and writers as well. Her story has appeared in poetry anthologies, biographical plays, documentary films, visual art exhibits on women in science. Her face, quiet, curious, compositas, become a symbol of timeless inquiry. One poet wrote, "She listened to corn, and the corn revealed a universe of music in green and golden code." Lessons from the cornfield. Barbara's teachings continue to guide research, ethics, science, policy, and even the framing of artificial intelligence. Her insistence that systems must be understood holistically, not just reduced into parts, has found echoes in ecological modeling, machine learning transparency, ethical engineering. She warned against arrogance and haste in manipulating life lessons that are more urgent than ever in a world of rapid biotech advancement. Her legacy offers a compass. Be curious, not just ambitious. Be precise, not just productive. Be open, not just clever. Immortal through impact. Barbara McClintock never wanted immortality. She wanted understanding. But in pursuing understanding with total devotion, she became immortal in the only way that mattered to her. Through the truth she uncovered, her discoveries changed the architecture of biology, inspired countless careers, expanded our view of what science is and how it should be done, and her example proved that greatness can come without acclaim, showed that science is as much about character as it is about knowledge. I never needed to be remembered, she said once. I only needed to know that I saw truly. And she did. Epilogue. A voice in the wind. In a letter left on Barbara McClintock's grave at Cold Spring Harbor, a young geneticist wrote, I came to science because of you. I stay in science because you remind me to listen. Barbara McClintock's life whispers to all who dare to seek knowledge with noise, but with presence, not with power, but with patience. She is the voice in the cornfield, the echo in the chromosome, the quiet certainty that even in silence, life is singing. If we listen, we will hear it too.